The Korres Homeopathic Production Unit is part of the Korres Natural Products Plant. It is the only authorized unit for the production of homeopathic medicinal products by the National Competent Authority. Additionally, it is certified for good manufacturing practices according to ISO standards for quality and for the environment. The homeopathic premises are characterized as a clean room area, meaning that the number of the airborne particles are controlled under specific limits through air handle units and high-efficiency performance filters. This way, the building monitoring system prevents the final product's contamination. Additionally, a water purification unit is established that produces purified water according to the current European Pharmacopoeia monograph. The PWNet includes a number of reverse osmosis filters and microbial removal filters. The value for water conductivity is lower than 1 microsiemens when the upper limit is 4.3. Regulatory status of homeopathic medicines. Homeopathic medicinal products cover a broad portfolio of safe, effective, and high-quality medicines mainly derived from natural substances. The authorization for a homeopathic medicinal product in Europe is regulated by specific provisions of directives on quality, safety, and efficacy. Homeopathic Pharmacopoeia and Production Guidelines. The main principles for the production of homeopathic medicinal products were set out by Dr. Hahnemann in 1810 in his book entitled The Organon of Medicine. Nowadays, homeopathic medicines are manufactured according to the German Homeopathic Pharmacopoeia, the current European Pharmacopoeia, and the Good Manufacturing Practices Guidelines. Compliance with pharmacopoeia monographs assures the safety and quality of the final product. Let's see the structure of a pharmacopoeia monograph through the example of the Ignacia remedy. The monograph Strychnos ignaci clarifies the botanical name of the starting material. Specifically in the monograph, the used plant part, dried ripe seeds, are clarified as well. The concentration of the active substances, such as alkaloids, strychnine and brucine, that are present in the original plant material and in the mother tincture. It gives the mandatory identification and assay tests for both plant material and mother tincture. The sector dosage forms clarifies the production method according to which the mother tincture is produced by the powdered herbal drug and subsequent liquid dilutions. In this example, the method clarified is the 4A. The incoming material has to be checked and released by the quality control team for further processes. The procedure takes place at a special design lab using specific analytical equipment like HPLC, UV, etc. The results are recorded on the raw material specification sheet. Based on the German Homeopathic Pharmacopoeia, 4 methods are mainly used for plant material, like the common remedies Lycopodium, Arnica, Ignacia. 1 method is connected to soluble materials, mainly in organic salts, like Natrium mutreaticum, Arsenicum album, Argentum nitricum. And 1 method is linked to insoluble starting materials, like Calcarea carbonica, Mercurius, Sepia. Potentization. The techniques of preparation of these drugs include the dilution of the raw material in hydroalcoholic solutions or in other excipients and the potentization of the product into different grades. Potentization is the process of vigorously mechanical strokes of the solutions, also called succussion. In most cases, the dilution is so high that it is almost impossible to find 1 molecule of the original raw material. There are mainly 2 dilution potentization scales, the centesimal and the decimal scale. The critical difference between them is actually the dilution ratio. In decimal scale, the ratio is 1:9, meaning that 1 part of concentrated substance is diluted in 9 parts of hydroalcoholic solution. For the centesimal scale, the ratio is 1:99 mass per mass. In both scales, the initial material for the first dilution step, based on the Pharmacopoeia monograph, is the concentrated mother tincture, or other potential substances. The second decendesimal dilution step is produced by the first decimal, following the ratio 1:9. In method 6 triturations of insoluble materials, the ratio 1:9 and 1:99 remains, using lactose monohydrate as the dilution vehicle. The centesimal scale was preferred by Hahnemann. The potentization process takes place up to the desired level of dilution grade, ranging from 12 to 50,000 times. The more potentized, diluted, is the starting material, the more active is the remedy. Let's see in practice the dilution and potentization phase. During Hahnemann's era, the potentization procedure was performed by hand. Nowadays, homeopathic producers use special designed equipment called potentizers. There are 2 types, the hand potentizer, multi-vials method, and the automatic single glass potentizer. The hand potentizer has been reliable for many years by doing an excellent job on potentization due to its counting device that stops the motor after a preset amount of 100 secessions. It is used in both scales, the decimal and the centesimal. The automatic single-glass potentizer machine is particularly used to produce Korsakov or K-dilutions following the techniques of Simeon Korsakov, who invented the method in 1829. The method is also known as a single-glass method and is applicable only to centesimal scale, where the dilution ratio is 1 to 99. Very high and reproducible dilutions would only become possible through this automatic machine, which method of preparation is similar to the classical Hahnemannian method. For further understanding, the single-glass potentizer produces high potentizations up to 50,000 dilution grade and 100 mechanical strokes between each dilution step. Documentation of the standard operation procedure ensures traceability of the starting material and excipients used, and it is in compliance with good manufacture practices requirements for pharmaceuticals. Impregnation. Impregnation is the process of spraying the potentized liquids into solid vehicles, mainly mixtures of sucrose-lactose globules. The process takes place also in special designed equipment. The basic model of the machine consists of two identical preparation stations, the modules, where different preparations can be made simultaneously. Each module has specialized designed glass receivers for containing the neutral globules to be impregnated. The application of the dilution on the grains is done through a unique sprayer that is screwed directly onto the bottle containing the required dilution. At the beginning of each preparation, the liquid flow has to be calibrated, and then the preparation process will run automatically. After spraying a calculated amount of the dilution onto the grains, the preparation will be homogenized inside the rotating receiver and dried automatically by thermostatic air. This process, spraying, impregnation, drying, will be repeated until the required dilution has been applied without any sticking of the grains. The dilute-jet system assures a perfect and reproducible transfer of the dilution liquid onto the grains. It also guarantees a homogeneous distribution of the correct quantity of the remedy on each individual grain. The quantity of dilution applied on homeopathic globules is set at 1% of their weight, in compliance with Pharmacopoeia Monograph Method 10, German Homeopathic Pharmacopoeia. The alcohol concentration of the potentized liquids should be at least 70% volume per volume. Documentation of the process is a part of the good manufacture practices, and it is mandatory for the traceability of the materials used and the record of the actual performance. Preparation of the requested galenic form. Encapsulation. The homeopathic remedies can be administered as liquid dilutions, impregnated globules for a range of sizes, or hard capsules of hypromellose. In case of hard capsules, the impregnated globules are filled in empty hypromellose capsules that are derived from plants. The process takes place at a special capsule filling machine station through a full documented process according to good manufacture practices. The net weight of the capsule is 170 mg. This process is controlled, and the line clearness is crucial for avoiding any microbial contamination. At the end of each batch preparation, the equipment has to be cleaned and autoclaved according to validated protocols. Microbiological control and storage of the finished products. The microbial limits for dosage forms are laid down in the European Pharmacopoeia Chapter 514 and should be applied to the final pharmaceutical product based on the route of administration. For the products for oral use, the water content is used to decide if the microbial limits for non-aqueous preparations or those for aqueous preparations, laid down in European Pharmacopoeia Chapter 514, are applicable. Researches indicate that the bactericidal effects of ethanol is a function that depends on the ethanol concentration and contact time. The content of ethanol in the remedy can therefore be used to decide if the microbial limits for non-aqueous preparations are applicable. The liquid potentizations should be stored in an alcohol concentration not less than 50% volume per volume. The potentized liquids for impregnation has to be freshly prepared by using at least 70% volume per volume alcohol concentration. The excipients like water, ethanol, sucrose globules, hypromellose capsules are tested for their microbiological contamination based on the requirements stated at their specific European monographs.